Hello, everybody, and welcome to today's Viva Biotech webinar, which is entitled Medicinal Chemistry for Cyclic Peptide Drug Discovery. During this webinar, we will share perspectives on the opportunities and challenges working with cyclic peptides, highlighting key considerations in the design, synthesis, and optimization. Whether you're exploring a novel peptide scaffold, or looking to enhance drug-like properties, we hope that today's webinar will provide you with some useful insights as to how medicinal chemistry contributes to the development of peptide therapeutics. Following the webinar, we will have a short 15-minute Q&A session, and I would like to encourage you to submit your questions via the Q&A box, which you'll see once the webinar commences at the bottom of the screen. My name is Simon Berry, and I will be the moderator for today's webinar. I'm the Vice President of Business Development at Viva Biotech and have worked in the global life sciences industry for the past 25 years. During that time, I've worked for contract research organizations based in the US, the UK, Australia, and China that provide a range of services spanning the entire drug discovery and development process, including chemistry, biology, admitox, and fully integrated drug discovery projects. Today's webinar will be presented by my colleague, Dr. Keja Ding. Keja is Executive Director of Chemistry at Viva Biotech and oversees a portfolio of medicinal and synthetic chemistry projects for a number of our North American and European clients. Keja has been a leader in Viva's chemistry team since 2015. His core scientific expertise is in medicinal and synthetic chemistry for structure-based small molecule drug discovery, which is enhanced by his understanding of molecular structures and interactions. In recent years, Keja has led chemistry efforts into the development of new drug modalities such as cyclic peptides, bifunctional molecules, and various drug conjugates. Keja received his PhD in chemistry from UCSD, and his thesis was focused on the design and synthesis of small molecules targeting viral RNA. I'd now like to hand over the microphone to Keja, who will present today's webinar. Keja, over to you.
Thank you, Simon. And I'm going to share the slide from my end so I can flip the pages. Okay, I think you guys can see my slide screen.
Yep, we got it.
All right. Okay, so let's get started. So today's topic is medicinal chemistry for cyclic peptide drug discovery. In today's talk, we're going to cover a few topics: cyclic peptide as drug modality, cyclic peptide drug discovery process. We'll see two examples. One is Cyclosporin A, and then the other one is MK0616. We'll look at the cyclic peptide chemical synthesis, and then towards the end, we'll have a Q&A section. So cyclic peptide as a drug modality. On the screen, we can see from the left-hand side, we have the traditional small molecule inhibitors. For example, the one we show on the screen is the AZD 0780, which is a PCSK9 small molecule inhibitor, and its molecular weight is 414, which is a typical sort of molecular size for small molecule drugs. On the right-hand side of the screen, we can see an antibody drug, which is much larger, 36,000 times larger than the the small molecule. And in between, we see a few different we consider new modalities. One is macrocycles. Still look like a small molecule, but it's a much larger compound in size. And we have in the middle the cyclic peptide we're going to talk about today. And then on the right-hand side, we also have the linear peptide, which is we show on the screen is the famous semaglutide, which is again, a much larger molecule compared to the MK0616 we're going to talk about today. So new drug modality of interest in recent years also include RNA, ADC, TPD, molecular glue, RDC, etc. But we are going to focus on the cyclic peptides in today's talk. So the reason for cyclic to go to cyclic peptide as a new drug modality is really to try to fill the gap between the small molecule and the larger biologics drugs. So hoping to bring the benefits and advantages from both ends and try to find a middle ground from the cyclic peptide. I think the main interest is in the target class, for example, the receptors and the protein-protein interactions that cannot traditionally be covered by small molecule drugs. And also to find oral bioavailability, which is not typically accessible by antibody drugs. Challenges for cyclic peptide include the again, oral bioavailability is not easily provided from the hit compound, and we're also need to overcome the stability issues and also find development strategy to guide us through the whole process. The cyclic peptide drug discovery process is very much like the other drug modalities. We have the hit ID process. More specifically for cyclic peptide, we have phage display, mRNA display, DL, and also de novo design by the AI. From hit to lead, and then ultimately to the preclinical candidate, we are going to use some of the methodologies that we also use for small molecule drug discovery. For example, the structure determination, SBDD, chemical synthesis, binding SAR, atomy property experiments and optimizations. But there are also unique methods and tools and strategies that we're going to need for cyclic peptide drug discovery. For example, it's a very different chemical space when we talk about the peptide bound, and also a few different optimization strategy. For example, cyclization, natural and non-natural amino acid, backbone modification, side chain modification, and also very important, the how we approach such complex molecules using chemical synthesis methods. The development of the cyclic peptide is encouraged by the advancement of a few technologies in recent years. This slide shows the hit discovery technologies that allow the discovery of a hit compound. So basically providing a starting point for the medchem development process. So we have phage display, mRNA display, DEL, and AI-based methods. They all cover pretty large chemical space, but they also have some unique features and differences. For example, the phage display typically cover natural amino acid only, and our mRNA display can cover some, but not that many non-natural amino acid. DEL is more compatible with the small molecule type of units. And then AI basis, of course, is not limited by the natural or non-natural amino acids. But they also have limitations that's related to the fundamental technology. Phage display, of course, is the phage-related limitations. mRNA is the RNA-related. DEL, of course, is the DNA synthesis and other related issues. For the AI-based, I think there is a lot. The main issue is there needs to have a much larger experimental throughput to validate the large number of hits from the program. Phage display and the DL screening are available at Viva Biotech. Now, the other important technology to allow cyclic peptide development is the structural determination and prediction. On the left-hand side, we have a couple examples. Both structures are from Cyclosporin A. One is the NMR structure, and then also the X-ray structure. In the middle, we have the MD simulation for various environments to simulate the conformation in a dynamic in a conformation for Cyclosporin A. On the right-hand side, of course, is the most recent sort of deep learning based methodology for structure prediction, including the cyclic peptides. So using the AI methods, we can do structural prediction, we can do sequence redesign, and then de novo design of the cyclic peptide. Viva Biotech has the capability to do all these structure determination experimentally, and also the by the AI program. So we're first going to take a look at the Cyclosporin A as an example of cyclic peptide. Cyclosporin A is a natural product. It is a calcineurin inhibitor, and its function as a immunosuppressant. So on the slide, we have a ChemDraw structure of Cyclosporin A, which is not that big. It has a molecular weight of 1200 and a C-log-P, calculated C-log-P of 14.4. TPSA is 279. So for many chemists who work in the small molecule space, is going to have a prediction of some of the properties related to structure like this. If we use the thinking from the small molecule side. So it's a very large small molecule, very hydrophobic. We expect good binding towards the target. The solubility is probably not going to be good. There's going to be high metabolism because of the it's a peptide in nature. Permeability is not a sure thing. Now, if it's a very hydrophobic molecule, we expect it to have a good passive permeability. But generally speaking, I don't think it's a very easily predicted property. Bioavailability, just by looking at the compound, I'm going to say because of its molecular weight, it tends to be low. But it's not the case for Cyclosporin A. This compound has an oral bioavailability of approximately 30%, which is very high for a cyclic peptide. And then in a different form of ChemDraw structure we show on the screen on the right, now we have a different sort of breakdown or highlight of some of the structural features of the Cyclosporin A. First of all, we noticed on the backbone amide ring, there are 7 nitrogen that is methylated. There's 1 D amino acid, the D-aniline, showing in the orange. We have 4 intramolecular hydrogen bonding of this compound that's based on the crystal structure, by the way. And then there's 2 non-natural amino acids highlighted in the pink color. Now, in the following slide, we're going to show some of the crystal structure. We're going to show some conformation flexibility and permeability. we're going to show some SAR studies based on the cyclosporin A, try to understand what's really going on with this cyclic peptide. So first, let's take a look at the crystal structures of cyclosporin A. There have been a good number of crystal structures published so far. On the slide, the top row shows the crystal structure of cyclosporin A alone. The difference is they are crystallized from different solvents. So from the left, we have the carbon tetrachloride, acetone, acetone, and water. One's crystallized from oil, ethanol, and surfactant. The fourth one is from the ether. These compounds, they have you can see the overall structure is sort of like a beta sheets. That's like two parallel sort of strands of amino acid that's really close to each other. Now, the top right compound is a crystal structure obtained from methanol and water. It's a fairly recent structure. And we can see significant difference from this structure to the ones that we see on the left. The polar environment has changed the crystal structure from the more like a beta sheet-like structure to a more open kind of conformation. So the ring is more relaxed and expand towards towards the outside. So the bottom row of the structures we show here are all from the co-crystal structure of cyclosporin A with a different protein. On the left, we have the co-crystal structure with whole human cyclophilin with cyclophilin from the T. gondii, a microorganism. And then we have the FAB and the RGG2 receptor, and on our right-hand side, we have transporter. The overall structure for these co-crystal structure, the cyclosporin A, all adopt a more open conformation, except for the last one, which still looks like a beta sheet. And if you notice, I have put a blue color sort of circle on the top left of each structure, or some of them. There is a amide bond in these structures that can switch between a cis configuration and a trans configuration. Now, if we look more closely, then we notice all the cis configuration structure are the more beta sheet-like structure, while all the trans amide bond configuration structure are all the open conformation. So there is certainly clearly a structural dynamics that's going on for cyclosporin A. And it looks like it has a lot to do with the hydrophobic versus hydrophilic environment that cyclosporin A is in. And this observation has been repeatedly investigated in the past. And some hypothesis or theory has been developed to explain the high bioavailability or the high cell membrane permeability of cyclosporin A. So the general hypothesis is in the more hydrophilic environment, the cyclosporin A would adopt an open and all trans conformation. Well, when it needs to pass the cell membrane, which the lipid bilayer is a more hydrophobic environment, then it will transform into the closed conformation, like the beta sheet-like conformation, and then get into the lipid bilayer, and then eventually pass through it. Now, the next question we have is, can we use cyclosporin A as an example and be able to set up some prediction parameters or empirically predict whether a cyclopeptide is going to be able to permeable through the lipid bilayer or not. And here we show a couple examples of close analog of cyclosporin A. Basically, the changes are, for example, cyclosporin G, we have a 1 extra carbon at this bottom position here compared to the SO from cyclosporin A, we have a propyl. For cyclosporin H, we have a D amino acid valine at this on the left-hand side of the chemical structure. And surprisingly, in the crystal structure of these compounds, cyclosporin G and H, both are crystallized from the hydrophilic solvent methanol. Cyclosporin G still adopts a fairly similar 3D structure like cyclosporin A, while cyclosporin H, due to one change in the chiral center on one of the amino acid, we're seeing a significant shift of the overall conformation. So basically from a cyclic open structure to a more convoluted and also called a clamp-like structure. So the backbone of the structure is sort of bent over. So the prediction of the 3D conformation of cyclic peptide is non-trivial. So next we'll take a look at some of the early SAR studies on the cyclosporin A, where more side chain modifications have been employed. And from this SAR table, and on the top right, we have a co-crystal structure with cyclophilin. And we can see if we modified R2 and R3 position to introduce more hydrophobic residues on the side chain, we can potentially increase the in vitro binding affinity of cyclosporin A with its target in a ELISA assay. However, this is not the same as the cell activity. Basically, we're seeing the cell activity is decreased. So this is a clear indication that altering the hydrophobicity or the side chain substitution, we're altering the binding in the eventual binding affinity, but we are also lowering, possibly lowering the cell permeability of the structure. In a different study, we're seeing more variations on different positions of the cyclosporin A. But in general, we are seeing that some of the modifications, if it's not at the interface of the cyclosporin A and a protein binding interface, then it's somewhat tolerated. However, we're seeing a non-consistent trend or a very difficult to predict trend of these compounds in a cell permeability assay like CACO2 and PAMPA. The calculated A-log P values for these compounds are rather very similar. And the author also performed a mouse PK study for cyclosporin A and a cyclosporin O. And we're seeing some differences in the PK profile. Now, in the original study, there is the explanation that cyclosporin A is going to accumulate in red blood cells, so that reflect as a lower plasma concentration. So I think the message we're getting from the study is small changes may result in big difference in permeability. And the binding affinity alone, especially the in vitro binding affinity, does not reflect the overall activity, the cell level activity of the cyclic peptide. So over the years, there are many statistical analysis of compounds that are beyond the rule of 5. So larger compounds, including the cyclic peptide. But I think overall there is still a limitation in terms of data, the availability of data. So data is not enough to really provide a very good guidance towards how we should predict or develop a cyclic peptide drug. In this case, I think try to focus on the experimental data collection and use it as a guidance for the development. Next, we're going to take a look at an orally bioavailable cyclic peptide drug called MK0616 or enlicitide. It is also an inhibitor of PCSK9, which is a tricyclic structure that can be dosed orally. Yes, with a permeability enhancer, Labrasol. The bioavailability is still considered low, less than 5%, but this is pretty good considering the T-half to be 35 to 130 hours, depending on the condition, the formulation. The compound is also very effective, seeing the PCSK9 reduction greater than 80% and also LDL-C reduction at approximately 65%. The in vitro binding affinity, the LDLR frat assay is showing a 5 picomolar IC50 or Ki value. Now, where this compound comes from? So it comes from a mRNA hit screening as a linear peptide, which is then cyclized to what we call the cyclic hit number 2, which comprises of a 12 amino acid with a Ki value of 956 nanomolar initially. So we're going to look at a more detailed MEChem development of this compound, but in general, through MEChem optimization, there's a key sort of preclinical ready compound, compound 44, that reach oral bioavailability at a 2.9% in cyno monkey with 7.6 hour T-half. And it's also very effective. Now, there's additional optimization from compound 44 to the final MK0616. And the optimization is mainly the synthesis, stability, tuning, and also solubility. So from the cyclic hit compound 2, the first round of MEChem optimization is from removing the exocyclic amino acids. And from the Ki values, we can see that the binding affinity is actually increased significantly by removing those residues. Here, the compound 4 was taken and checked for GI protease activity, whole blood T-half, and also a mouse PK study. So we can see the red colored digits in this chart reflect some of the not so stable properties of this compound. 2 protease inhibition at a very high level. In a mouse whole blood, it's 1 hour half-life, and in a cyno monkey, it says it's not determined. So taking the compound 4, there is a sort of alanine D-amino acid and backbone nitrogen atom methylation scan. This is actually a very important study. So walk around all the 9 amino acid that's in the link that's in a cycle, and replace every residue with a smaller side chain substitution. So the aniline, just leave a methyl group there and then see if that change is tolerated. Now, if it is, that basically suggests the compound, that side chain is not forming any key interaction with the target. The D-amino acid is also investigated. So basically seeing about the spatial characteristics for different positions. And also the unmethylation, which is a very typical sort of cyclic peptide modification strategy to reduce the hydrogen bond donor capability to potentially then address the both the stability and also the permeability property is investigated. Now throughout we can see the aniline replacement resulted in position 1, 3, 5, 6, and 8. All these positions tolerated the modification, the alanine replacement. And we'll see in a later study, these positions are heavily explored and modified to finally achieve the drug molecule. Now there is a red arrow here, that is the glycine 2. In a later study, we'll see that this position is actually very important, and the L amino acid modification is not tolerated, but this position tolerates a D amino acid. Now, in this initial scan SAR study, this position is not included, but it will be included in a later slide. So at this point, we have a very good early understanding of the SAR of this hit compound. So we highlight in this ChemDraw structure the pink color, also including the backbone nitrogen atom. So the pink color indicates there is potentially key interactions from these residues or NHs. And the green color indicates the good tolerability for replacements, and likely suggesting there's no key interaction from these structures residues. So based on this, what we wanted to see is how are we going to modify this compound further, and then to improve the binding, and then to improve the stability and also permeability. So in the later slides, we will see D and L modifications for glycine 2 side chains. We'll see confirmatory modifications for the backbone NH and the side chain OH and NHS. We'll see modifications including non-natural amino acid with linker modification. We'll see the modification on the amide backbone, and also we'll see crystal structures that guide the structure-based drug discovery process. So in this slide, on the top row, we see the proline 8 modification. This position, when modified to aniline, there is some loss of activity, but it's not that significant. So we're seeing further modifications here. And notice this compound 30, which almost bring a 10-fold increase in the binding affinity. This structure is also included in the final compound. So this is the where this modification is presented. At the bottom left, the 5-fluoro-tryptophan 4 modification. So removing the fluorine atom actually decrease the binding affinity by 10-fold. So that fluorine is actually important. In the 5-fluorotryptophan 3 modifications, we're seeing some ring changes, removing the NH from the tryptophan and also some bi-aryl system, or open up the indole ring. And in this case, we're seeing that, first of all, the nitrogen atom is not critical for binding. So replacing to the naphthalene is it can maintain the binding affinity. So it's mostly hydrophobic driven binding at this position. Compound 30 was taken to check the GI protease activity, whole blood stability, and the mouse PK. And we use green color to highlight the part where the stability is improved. So we can see one protease elastase is improved, and also the mouse blood T half is improved to 311 minutes, and the cyno monkey whole blood is greater than 360. So the overall stability of this compound is better. And a co-crystal structure of compound 30 is obtained. And from the structure, we can see there are 5 key hydrogen bonding, all from the backbone NH and the carbonyl groups. We're seeing the floral tryptophan 3 and 4 occupy hydrophobic pockets on the top left and the middle of the binding site. The proline, the methylated proline 8 and the 777 has this sort of twisted structure of the backbone, which will be a very important structural feature that's going to appear in a later slide. And also we notice that glycine 1, the lysine 1, glycine 2, aspartic acid 5, and histidine are all solvent facing. And these residues can potentially be further modified. Taking compound 30, in this slide, we're seeing some modifications, especially including the removal, phosphol, the removal of this orange color sort of exocyclic functionality leads to a maintenance of the binding affinity, and then adding a D-aniline at a 2 position, again, maintain the binding affinity. So they're suggesting there is a space around this position. And also capping the tyrosine on the right-hand side using a methyl group does not affect the binding affinity. So there is no hydrogen bonding interaction of this OH. Compound 40 is co-crystallized with the target. And I think one key feature is, again, confirm the 2-position D amino acid is tolerated and it's solvent facing. Also, we see some additional improvement in the protease and whole blood profile from these modifications for compound 40. And taking compound 40, there is another round of modifications on the 2 position, all D amino acid. And we can see in some cases where mostly the hydrophobic all carbon residues, the binding affinity is maintained, while introducing a polar group. There's a loss of the binding. Modification of aspartic acid 5 and histidine 6 is shown in this slide. And in some of the cases, we can see reducing the cyclization of the aspartic acid and also reducing the histidine to a smaller amino acid residue, maintain the binding. And this basically confirms the observation in the crystal structure of compound 30 that these positions do not form direct interaction with the target. Co-crystal structure of compound 51 is obtained. Now in this crystal structure, there is a key feature that we present using the red arrow, that there is close proximity between some of the side chain residues and one below it, between the tryptophan and also the linker. So later we'll see this crystal structure is utilized as evidence for structure-based drug design to cyclize the compound and ultimately forming a tricyclic structure. In this slide, we see some optimization of lysine 1 because there is some observation that the compound may have some mast cell degranulation activity. So this activity is not desired. And from this SAR, we can see mainly that if we cap the right-hand side tryptophan using a messenger group and introducing the two-position aniline, then we're getting the negative result from this activity. In this slide, we're seeing the first compound, 78, that is cyclized at the top position to form a bicyclic structure. The binding affinity increased from 11 nanomolar to 1.5 nanomolar. And stability also looks pretty good, except for this chymotrypsin protease activity. And the other interesting thing is probably what the chemist has expected, that the overall conformation, especially the backbone conformation of this cyclic structure, is not changed. Upon introduction of the second ring structure at the top. This explains the maintenance of the binding affinity. So here we look at the chemical synthesis of the bicyclic structure. So starting from a linear peptide, 76 on the left-hand side, that is from the solid phase synthesis. So the author first introduced the bicyl ether cyclization at the bottom position, and then followed by a amide bond formation at the second ring cyclization. Once the bicyclic structure is formed, the author then investigated the modifications on the newly formed ring, explored the size, the effect of the size of the ring, and also some other modifications. So overall, we think it is clearly shown that compound 78 is still the optimal structure with the highest binding affinity. Compound 84 is another interesting compound because it changes this 5-fluorotryptophan to a structure without the NH functionality. So again, further confirm that this binding pocket is mostly hydrophobic driven. So once the bicyclic structure SAR is done, the author introduced a different cyclization strategy on the second ring that's at the top. So from the amide bond to an olefin cyclization, which then increased the binding affinity by approximately 3-fold. The formation of the additional ring between the tryptophan in the middle and then the linker phenyl ring at the bottom significantly increased the binding affinity by approximately 47-fold. The crystal structure of compound 35 is obtained. And again, one interesting thing is the overall conformation of the large cycle is unchanged. There is some flexibility in a bottom linker ring, which we highlight as green. The flexibility of this ring is contributes to the maintenance of the overall binding. Again, the chemical synthesis of these ring structures. So the top row, the top scheme shows the cyclization strategy for using olefin metastasis. Again, the bottom ring is formed first, the bicyl ether, and then followed by the top ring cyclization using the olefin metastasis. The bottom scheme shows the cyclization of the tricyclic structure. So again, starting from the compound 32 as a linear peptide, but with additional functionalities included for cyclization. So first, again, the dicyl ether connection is made, followed by the click chemistry to form the trisyl ring in the middle. And then the last is the alpha metastasis at the top. There can be different cyclization strategy for different compound design, but this is what the author used for these compounds. So both the bicyclic and the tricyclic peptides are further optimized on the side chains, which eventually leads to the key compound 44 that is picomolar potent binding affinity with fairly good oral bioavailability. That's clinical ready. The next slide shows the chemical synthesis for the actual MK0616 that enters the clinical trial. Now, from compound 44, the author describes the optimizations mainly for the chemical synthesis to further improve the stability and the solubility. But for MK0616, the overall synthetic complexity is still significant and involve many steps of building block non-natural amino acid synthesis and also the final ring formation. However, if we take a look at this compound retrosynthetically and then break it down step by step into the smaller building blocks, we can clearly see a modular approach of the synthesis, and that can be managed with careful planning and strategic considerations from the medicinal chemists. So it's complex structure at a first glance, but again, careful planning and optimization along the development process can overcome such complexity. Unimodular approach and a ring cyclization strategy, and then balancing synthetic feasibility with design priorities. So cyclic peptide synthesis is not new. There have been many published examples and the compound that's currently in the lab being made. There are many different cyclization strategies. For example, from the simplest disulfide bond, various cyclic ethers can use microaddition as cyclization method, amide bond formation triazole via click, and ring clothing metastasis. There can also be side chain modifications or modifications in general on the backbone, non-natural amino acid, stapling of the alpha helix, PEG modifications and linker strategy, fatty acid attachment, phosphorylation, biotin labeling, and fluorescent labeling for tool compounds. There can also be conjugation of the cyclic peptide structures to other molecules, for example, antibody and a radioactive compound. So overall, there have been very successful examples in a large number of peptide, linear peptide, cyclic peptide, and a more complex peptide that's being made in labs across the globe, and also in the Viva chemistry labs, using both old and new synthetic methodologies and also modern instrumentations. It's really a combination of these methods and tools that allow us to do efficient cyclic peptide and a peptide synthesis in general. All right, so I think that is the last slide that I have for today's.